Hello there, and welcome back to the Sleepy Science Documentary Podcast. Tonight, we're exploring a question that sits at the boundary of science fiction and genuine scientific breakthrough. Could scientists really bring extinct animals back to life? We'll journey through laboratories where geneticists are rewiring the very code of life itself, through permafrost, where ancient DNA sleeps beneath ice, and into the ambitious dreams of companies betting billions that the dead need not stay dead. This is not the fantasy of resurrection, but something far more intricate. It's the precise, patient work of editing genes, sequencing genomes from fossil fragments, and carefully crafting new versions of creatures the world has lost. Some of these animals vanished thousands of years ago, their bones scattered across museums and frozen ground. Others disappeared mere decades ago, leaving behind frozen tissue and preserved museum specimens that hold the secrets of their genetic code. If you enjoy these slow explorations of how science bends the boundaries of what we thought possible, I invite you to like, subscribe, or share a thought below. It helps others find their way to these quiet corners of wonder. But for now, just settle in, let your breathing slow, and allow yourself to imagine a world where extinction might not be forever. Let's begin. De-extinction, when you first encounter the term, sounds like pure fantasy. The word itself suggests bringing something back from death, resurrecting a creature that walked the earth millions of years ago and is now gone forever. But the actual science is far more nuanced than the term suggests. De-extinction is not about creating perfect genetic copies of extinct animals from the distant past. Instead, what scientists are doing is developing what they call functional proxies, modified versions of living animals that carry some of the traits and ecological characteristics of their extinct relatives. When researchers talk about bringing back the woolly mammoth, they are not describing a true resurrection, they are describing the genetic editing of an Asian elephant to possess certain mammoth traits: a shaggy coat, subcutaneous fat layers, and cold tolerance adaptations. The resulting animal would be neither purely a mammoth nor purely an elephant, but something genuinely novel, a hybrid engineered to fill an ecological role that has been empty for thousands of years. This distinction matters enormously. The language of de-extinction can be somewhat misleading, and many scientists themselves acknowledge that what they're creating is not true resurrection, but rather a carefully designed approximation. Even the founders of major de-extinction companies admit that the terminology is deceptive. They are not bringing extinct animals back from the dead in any traditional sense. They are engineering new organisms that approximate the form and function of their extinct predecessors. The term has become popular in media and marketing because it captures the public imagination, but it obscures the actual technical achievement. What's happening in laboratories around the world is genetic engineering of unprecedented precision and ambition, a process that requires understanding not just what an extinct animal looked like, but how its body functioned at the genetic level. The woolly mammoth has become the flagship project for de-extinction research, the animal that captures most media attention and represents the ultimate goal of companies like Colossal Biosciences. Unlike dinosaurs, which vanished 66 million years ago, leaving only fossilized bones, woolly mammoths died out relatively recently, around 4,000 years ago in the most remote Arctic regions. This recency means that permafrost has preserved actual DNA from mammoth tissues, creating a genetic blueprint that scientists can now access and study. Colossal has announced that it is on track for embryos to be ready for implantation by the end of 2026, with hopes of a calf being born in 2028. To achieve this, the company has edited 25 genes so far, and expects to ultimately edit around 85 different genes, some with multiple functions related to cold tolerance. The genetic transformation is vastly more complex than earlier de-extinction attempts, because woolly mammoths evolved over hundreds of thousands of years to survive in Arctic conditions. Each gene they edited codes for specific traits, some controlling hair growth, others influencing fat deposition, still others regulating body size and metabolic rate. The approach combines CRISPR gene editing technology with genetic material from Asian elephants, the mammoth's closest living relative. By comparing preserved DNA fragments from permafrost with elephant genomes, scientists identify which genes are responsible for distinctive mammoth characteristics. These genes are then inserted into elephant embryos, creating animals that carry both elephant and mammoth genetic instructions. The process is not instantaneous. Creating each edited embryo requires precise manipulation of elephant eggs, introducing the genetic modifications, and then monitoring development. Scientists have had to refine the techniques repeatedly, learning from failures and successes how to most effectively create stable embryos that can develop to full term without the sort of defects that killed the cloned Bucardo decades earlier. DNA preservation in permafrost represents one of the great fortunate accidents of geology and paleontology. When animals died in the Arctic regions and became trapped in frozen ground, the extreme cold essentially halted all biological decay. Bacteria and fungi that normally break down tissues cannot function in temperatures far below freezing. Permafrost acts as a natural freezer, maintaining tissue integrity for thousands of years. Scientists have recovered bone, skin, hair, muscle tissue, and even blood from mammoths and other Ice Age creatures preserved in these frozen tombs. The mammoths of Siberia are particularly valuable because some specimens contain enough preserved soft tissue to yield multiple samples of DNA. A single mammoth tusk or femur can be sampled multiple times without prey, destroying the fossil. The key advantage of permafrost preservation is that it avoids the chemical damage that occurs in other preservation methods. When bones sit in sediment at normal temperatures, groundwater slowly leaches minerals and organisms gradually degrade proteins and DNA. But in permafrost, these processes essentially pause. The cold has kept tissues in a state of suspended preservation for millennia, allowing modern scientists to extract genetic material that is thousands of years old. The challenge now is that even preserved DNA degrades slowly over time. The longer something sits frozen, the more fragmented the DNA becomes. But modern sequencing technology can stitch these fragments back together, like reconstructing a broken ancient manuscript by finding overlapping pieces. Scientists have found that the best preserved specimens tend to be found in regions with the most stable and consistent cold, where temperature fluctuations are minimized. In these ideal conditions, mammoth DNA has remained extractable for tens of thousands of years. Ancient DNA degrades in predictable patterns. When scientists examine genetic material from fossils and preserved specimens, they find that DNA breaks into shorter and shorter fragments over time. This fragmentation follows a measurable pattern based on how long the specimen has been preserved, what temperature it was stored at, and how much biological activity occurred before preservation. For mammoths specifically, scientists have found that they can reliably extract DNA fragments up to several thousand years old. The oldest woolly mammoth DNA yet recovered comes from specimens that died around 50,000 years ago, and even after that span of time, fragments remain retrievable. The DNA that survives is typically in pieces ranging from 30 to 100 base pairs long, whereas modern DNA in living animals consists of complete strands millions of base pairs long. Scientists must assemble these fragmentary pieces like an enormous degraded puzzle, using computer algorithms that compare the fragments against known genomes of living elephants. The fragmentation pattern is predictable enough that researchers can determine authenticity. DNA from contamination, from modern humans handling the specimen, from bacteria colonizing the bone, shows different damage patterns than ancient DNA. This helps scientists distinguish between genuine mammoth DNA and environmental contamination. The process reveals not just what the DNA sequence is, but also how it has been altered by time. Some bases within the DNA undergo chemical changes that leave specific signatures of age. CRISPR gene editing represents a revolutionary breakthrough that fundamentally changed which animals might realistically be brought back. CRISPR stands for clustered, regularly interspaced short palindromic repeats, a system that bacteria naturally use as an immune system against viruses. Scientists harnessed this system by using a protein called Cas9, which acts like molecular scissors. These scissors can cut DNA at precisely targeted locations, and when the cell repairs the cut, scientists can manipulate what goes into the repair. They can remove genetic sequences, add new ones, or change existing instructions. What makes CRISPR revolutionary is its simplicity and accessibility compared to previous gene editing technologies. Before CRISPR, genetic engineering required complex equipment and rarely achieved high accuracy. CRISPR, by contrast, is relatively inexpensive and can be delivered into cells using modified viruses or other vectors. The elegance of the system is that scientists only need the DNA sequence they want to edit into. They do not need living cells from the extinct species. With fragmentary DNA sequences from fossils, they can map out the genetic differences between a living species and an extinct one, then use CRISPR to edit the living animal's DNA to match the extinct species' genetic code. The process involves creating embryos with edited genomes, allowing them to develop in the laboratory, and then implanting them into surrogate mothers to bring them to full term. The technology has advanced so rapidly that what seemed impossible a decade ago is now becoming routine in research facilities worldwide. Multiple de-extinction companies are now using CRISPR as their primary tool, having abandoned cloning approaches as too impractical for most species. The passenger pigeon initiative began in 2012 with a clear and ambitious goal: to restore a species that once numbered in the billions and was hunted to extinction within just a few centuries. Before European settlement of North America, passenger pigeons were the most abundant bird species on the planet, and their flocks were so vast that they would darken the sky for days as they flew overhead. Their extinction was remarkably swift, driven by commercial hunting that treated them as an infinite resource. By 1914, the last wild passenger pigeon was dead, and the species vanished completely. The Great Passenger Pigeon Comeback Project seeks to recreate this bird, not to recreate the past, but to restore the ecological role the bird played in North America's eastern forests. Because cloning does not work for birds, the approach differs from the woolly mammoth project. Scientists plan to collect primordial germ cells from band-tailed pigeon embryos, cells that have the potential to become eggs and sperm. These cells can be genetically edited to carry passenger pigeon genes, and through techniques still being refined, these cells can be grown in laboratory conditions to eventually produce eggs and sperm that carry passenger pigeon genetic material. Initial timelines suggested completion by 2025, but scientists have since adjusted these predictions. Current estimates suggest that recreated passenger pigeons might hatch between 2029 and 2032, with the possibility of a thriving aviary population by the 2030s. The revised timelines reflect the genuine complexity of avian genetics and reproduction, areas where scientists have less practical experience than with mammalian systems. Working with birds requires understanding entirely different reproductive mechanisms and hormonal systems. Museum specimens represent an often overlooked treasure trove of genetic material. When naturalists in the 19th and early 20th centuries collected animal specimens, they often preserved them in alcohol or formaldehyde, or they dried and mounted skins for display. These specimens, stored in museum collections around the world, contain preserved tissues that still hold extractable DNA. Museum specimens have particular value for recently extinct species because the preservation was done deliberately, with care taken to maintain tissue integrity. A taxidermied animal skin, properly stored, can retain usable DNA for centuries. The passenger pigeon project relies heavily on museum specimens because thousands of passenger pigeons were collected and preserved in natural history museums across North America and Europe. Similarly, specimens of other extinct birds and mammals provide the genetic blueprints that scientists use to reconstruct vanished genomes. The oldest extractable DNA from a museum specimen comes from the great auk, an extinct seabird that was hunted to extinction in the 1840s. Researchers have successfully sequenced DNA from specimens that are over 150 years old. Each specimen adds layers of genetic information. By comparing multiple specimens, scientists can identify genetic variation within the species, understanding not just what the species was, but the diversity that existed among individuals. This information is crucial for recreating a species that would have reasonable genetic diversity, rather than being composed of clones or near-identical copies. Museums are becoming increasingly recognized as repositories of genetic heritage, places where the biological legacy of vanished species rests in temperature-controlled rooms, waiting for technologies that can unlock their secrets. Cloning and genetic engineering represent two fundamentally different approaches to de-extinction, each with distinct advantages and limitations. Cloning, the technique that created Dolly the sheep in 1996, involves taking the complete nucleus from a cell of the extinct species and inserting it into an egg cell that has been emptied of its own nucleus. The reconstructed egg can then be stimulated to begin development, creating an animal that is genetically identical or nearly identical to the source individual. Cloning requires a living cell, or at least a very well-preserved cell, from the extinct species, which severely limits which animals can be revived using this method. The Pyrenean ibex, a subspecies of wild goat that went extinct in 2000, was briefly brought back through cloning in 2003. A single preserved skin specimen from the last ibex provided enough genetic material to clone an animal, but the cloned kid was born with severe complications and died within minutes. Genetic engineering, by contrast, uses CRISPR or other gene editing tools to modify a living species to approximate the genetic profile of an extinct one. This approach requires only DNA fragments, not intact cells. The advantage is that it opens possibilities for ancient species where no well-preserved cells exist. The disadvantage is that genetic engineering creates something new rather than a true copy of the original species. The Pyrenean ibex, or Bucardo, lived in a single mountain range between Spain and France until the early 2000s. The last wild individual was shot in 2000, though a few preserved specimens existed in museums. Scientists made several attempts to clone this animal, with one project succeeding in producing a living clone in 2003. The cloned Bucardo was gestated in a surrogate mother, a domestic goat, and the clone was born alive. However, the animal had severe lung defects that had not been anticipated, and it died within minutes of birth. The incident highlighted both the potential and the profound challenges of de-extinction through cloning. The genetic material from the preserved specimen had been damaged by time, and the decoding of that damaged DNA had led to errors in the reconstructed genome. The clone's body could not function properly. Despite the tragic outcome, The Bucardo remains the only extinct animal to have been brought back to life, even briefly. The attempt provided invaluable lessons about what could go wrong and what scientists needed to address in future projects. Genetic defects, developmental errors, and physiological incompatibilities between the new genome and the surrogate species biology all created obstacles. These lessons shaped how later projects, like the Woolly Mammoth Initiative, are being designed with greater care and more comprehensive genetic analysis. Surrogate mother species selection is crucial to any cloning or embryo transplantation project. For woolly mammoths, scientists are using Asian elephants, the closest living relative. Asian elephants have the longest gestation period of any mammal, about 22 months, which provides the time needed for a mammoth-elephant hybrid embryo to develop to full size and viability. The elephant's reproductive system is sufficiently similar to what scientists believe a mammoth's was, making the genetic programming compatible at a basic level. For the Tasmanian tiger project, scientists are using the fat-tailed dunnet, a small marsupial relative. For the passenger pigeon, the surrogate is the band-tailed pigeon, a close living relative. Each choice involves careful assessment of compatibility. The surrogate must be able to tolerate an embryo with partially foreign genetic instructions. The surrogate's body must be able to provide the right hormonal environment for development. The size of the mother must be compatible with the expected size of the developing fetus. These constraints limit which species can realistically be brought back. A large extinct species like a mammoth needs a large living relative as surrogate. Smaller extinct species might have more surrogate options. The process of selecting and breeding appropriate surrogates has become part of de-extinction research, with facilities maintaining populations of surrogate animals ready for implantation projects. The Tasmanian tiger, or thylacine, represents one of the most ambitious de-extinction targets beyond the mammoth. This carnivorous marsupial roamed mainland Australia, Tasmania, and New Guinea until modern times, when it was hunted to extinction. The last wild thylacine was shot in the 1930s, and the last captive individual died in a Tasmanian zoo in 1936. The recent extinction means that preserved specimens exist in multiple museums, and DNA recovery from these specimens has been remarkably successful. Colossal Biosciences announced that it had reconstructed the thylacine genome to over 99.9% accuracy, an extraordinary feat of genetic archaeology. The company even recovered a 100-year-old preserved head of a thylacine that had been skinned and kept in ethanol, and from that specimen they extracted RNA molecules, which are even more fragile than DNA, but provide additional genetic information. The fat-tailed dunnart, a small living marsupial, is being used as the template for editing. By inserting thylacine genes into the dunnart genome, scientists are creating a hybrid that would possess the distinctive features of the thylacine, including its carnivorous adaptations, its body shape, and its predatory behaviors. The project demonstrates how recent extinctions offer advantages because of better preserved genetic material. Ancient elephant species relationships provide context for understanding the woolly mammoth's place in evolutionary history. The modern Asian elephant shares approximately 99% of its DNA with the woolly mammoth, a genetic similarity that makes the elephant an ideal template for mammoth reconstruction. Scientists have traced the evolutionary relationships between modern elephants and extinct species by comparing their genetic sequences. The woolly mammoth diverged from the Asian elephant perhaps 4 to 6 million years ago, during which time the mammoth lineage accumulated genetic adaptations to Arctic life. These adaptations are visible in preserved mammoth remains, which show thickened skin, dense fur, large ears that have been reduced compared to modern elephants to conserve heat, and subcutaneous fat layers that help retain warmth. The genetic differences between mammoths and modern elephants amount to roughly 1% of the total genome, but these differences encode the specific traits that made mammoths suited to their environment. Understanding these relationships helps scientists identify which genes are most important to engineer. Rather than editing hundreds of genes with unknown functions, researchers can focus on the specific genes that code for recognized mammoth traits. The comparison of genomes reveals not just which genes differ, but also reveals something about the process of evolution itself, showing how natural selection shaped different species to fill different environmental niches. By studying the genetic changes that occurred as mammoths adapted to ice ages, scientists gain insight into which genetic modifications would allow modern elephants to similarly thrive in Arctic environments. This knowledge of evolutionary adaptation becomes the blueprint for genetic engineering. Extracting DNA from fossils is a technical challenge that has improved dramatically over the past 2 decades. The process begins with identifying samples likely to contain preserved genetic material, typically bone or tooth tissue, rather than skin or soft tissue. The sample is carefully cleaned to remove surface contamination, then ground into powder using sterile techniques. The powder is treated with chemicals that break down the mineral structure of the bone, allowing DNA to be extracted. The DNA that emerges from this process is highly fragmented, typically in pieces far shorter than the intact DNA found in living cells. These fragments are then sequenced using modern high-throughput sequencing technology, which reads the genetic code of millions of fragments simultaneously. Computers then assemble these fragments by finding overlapping regions, much like assembling a jigsaw puzzle. The assembly process uses reference genomes from living species as guides, leveraging the known genetic sequences to help position the fragments correctly. This process has been refined to the point where scientists can reconstruct ancient genomes with remarkable accuracy, even when 99% of the sample is contaminated with bacterial or fungal DNA. Their stories increases as specimens grow older because DNA degrades at a predictable rate, and very ancient specimens may have DNA in such small pieces that assembly becomes nearly impossible. Scientists typically extract DNA from multiple specimens of the same species, combining genetic information from different individuals to create a more complete and accurate reconstruction. This approach also captures genetic variation within the species, information that becomes crucial when designing de-extinct populations. The Quagga project in South Africa represents one of the earliest successful de-extinction efforts, though it predates modern genetic engineering. The quagga was a subspecies of zebra, native to South Africa, characterized by stripes that were less extensive than those of modern zebras, with plain coloring on the hindquarters. The last wild quagga was hunted to extinction in the 1870s, but museum specimens preserve the species' appearance. Scientists noticed that modern zebra populations in South Africa showed significant genetic variation, and some individuals occasionally exhibited stripes that resembled quagga patterns. Beginning in the 1980s, researchers selectively bred zebras that carried quagga-like traits, essentially breeding backwards through the zebra genome to recreate quagga characteristics. After multiple generations of selective breeding, they produced animals that resembled historical descriptions and paintings of quaggas quite closely. These animals, called quaggas, though technically zebras with bred-back traits, never achieved the genetic identity of true quaggas, but they demonstrated that effective burn. Selective breeding could recreate extinct morphologies. The Quagga project showed that de-extinction was possible using conventional breeding, though it required patience and large populations to work with. The project also illustrated the limitations of selective breeding. Genetic drift, The random changes in gene frequency that occur in small populations constrained the project's success. Modern genetic engineering, using CRISPR, promises to achieve quagga recreation far more rapidly and with greater precision than traditional breeding alone. Breeding back lost traits through selective reproduction has been used for centuries to reshape domesticated animals and create new breeds. When farmers select animals with desired characteristics for breeding, they are essentially performing artificial evolution, concentrating genes for those traits in successive generations. Scientists have applied this approach to extinct wild species by selecting living relatives that carry ancestral traits. For certain extinct cattle breeds, researchers have identified modern cattle that carry genes for characteristics seen in historical records or preserved remains. By breeding these animals selectively, scientists can increase the frequency of ancestral traits. The Auroch reconstruction project in Europe has used this approach with some success. The Auroch, a massive wild cattle species that went extinct in the 17th century, is being recreated through selective breeding of modern cattle breeds that carry Auroch genetic material. Some European conservation groups have conducted breeding programs that have produced animals with impressive size and aggressive temperament, matching historical descriptions of Aurochs. However, breeding back remains limited by the genetic diversity available in living populations. If a particular trait was lost from the breeding population, it cannot be recovered through selective breeding alone. This limitation is where genetic engineering provides an advantage. If the ancestral gene sequence is known, CRISPR can reintroduce it directly without waiting for it to reappear through random genetic variation. The Auroch reconstruction efforts represent one of the most visible examples of breeding back de-extinction currently underway in Europe. The Auroch was a massive bovine with bulls weighing over 1,000 pounds, wild and aggressive, with magnificent horns. Hunted extensively, the species declined until the last Auroch was killed in 1627 in Poland. European scientists have since identified modern cattle breeds that carry Auroch genetics, particularly certain primitive breeds from Eastern Europe that show genetic markers matching prehistoric Auroch remains. By selectively breeding these cattle, concentrating Auroch's traits through multiple generations, researchers have produced modern animals that closely resemble historical Aurochs descriptions and images. These reconstructed Aurochs are living in several European wildlife reserves, where they graze, breed, and behave like the wild cattle of prehistoric times. The project demonstrates that de-extinction need not always be high-tech. Sometimes, careful observation of what genetic material still exists in living populations, combined with strategic breeding, can resurrect extinct forms. However, genetic engineering may eventually be applied to accelerate the process or to recreate traits that have been completely lost from modern cattle populations. The project also raises questions about ecological reintroduction. Recreating the animal is one challenge. Returning it to an ecosystem where it has been absent for centuries is another, particularly when the ecosystem itself has been transformed by human activity. Genetic diversity requirements present one of the fundamental challenges to any de-extinction project. A species that consists of too few individuals or genetically similar individuals faces serious problems, including increased disease susceptibility, reduced fertility, and developmental abnormalities. Scientists aim to ensure that de-extinct species have sufficient genetic diversity to avoid these problems. For mammals like the woolly mammoth, genetic diversity can be increased by using genetic material from multiple individual mammoths, preserved in permafrost. DNA from different specimens allows scientists to identify genetic variation and recreate animals with diverse genotypes rather than identical clones. For species where only a few specimens remain, such as the passenger pigeon, scientists can extract DNA from multiple museum specimens collected at different times and places. representing individuals from different populations. The genetic diversity question also shapes how de-extinction projects are envisioned long-term. Scientists are not typically planning to create a single de-extinct animal, but rather to establish breeding populations with sufficient diversity to remain viable. Plans for the passenger pigeon project involve eventually creating a free-flying population in restored forest habitat, not keeping birds in captivity. The diversity needed to sustain such a population in the wild requires careful genetic engineering to ensure that the recreated species carries variation comparable to what the original species maintained. The gastric brooding frog attempt represents a more speculative de-extinction project. This Australian frog, which went extinct in the 1980s, had an extraordinary reproductive adaptation. The female would swallow her eggs, and the tadpoles would develop inside her stomach. The stomach would stop producing acid during this period, allowing the young frogs to develop safely before the mother would regurgitate fully formed froglets. The species disappeared so rapidly that scientists initially had no idea why. The attempt to recreate this frog raises unique challenges. Frogs require living cells for cloning more strictly than mammals, and no frozen frog tissue was preserved in time. DNA has been extracted from preserved museum specimens, but the sample is limited. More fundamentally, the complex developmental processes involved in amphibian reproduction remain poorly understood compared to mammalian reproduction. Scientists would need to not only recreate the frog's genetic code, but understand the hormonal and developmental systems that regulate its unique gastric incubation. The gastric brooding frog project demonstrates that not all de-extinction attempts are equally feasible, even with theoretical genetic knowledge. Some species have life histories and reproductive strategies so specialized that recreating them requires understanding developmental biology at a level not yet achieved. Synthetic biology approaches to de-extinction represent the most theoretically advanced methods under exploration. Rather than relying on cloning or even traditional gene editing, synthetic biology involves designing genomes from scratch using computer modeling and then synthesizing the DNA directly. Scientists can theoretically design an entire genome at the molecular level, write out the genetic code as a sequence of chemical instructions, and then synthesize that DNA strand by strand. This approach would theoretically allow de-extinction of very ancient species where fragmented DNA is the only source material. Instead of assembling fragments like a puzzle, scientists could reconstruct a complete genome computationally based on fragmentary evidence and knowledge of how genomes work. The DNA could then be synthesized and inserted into a cell to begin development. Synthetic biology remains largely theoretical for de-extinction purposes, but the technology is advancing rapidly. Scientists have already successfully synthesized entire viral genomes and even entire bacterial genomes, creating living organisms from scratch based on genetic designs. The application to de-extinction requires solving additional challenges, particularly understanding which genetic variations are essential to recreating a species' unique characteristics versus which variations are neutral. The approach also raises profound ethical questions about creating life forms that have never existed before based on limited historical genetic knowledge. The Bucardo clone that lived minutes remains the only extinct animal to have been brought back to life, even if only briefly. In 2003, Spanish and French scientists successfully cloned a Pyrenean ibex using genetic material from a preserved ear specimen. The clone was implanted into a domestic goat, and the pregnancy proceeded normally. The Bucardo was born alive, a living embodiment of a species extinct just 3 years earlier. However, within minutes, the clone died from massive lung defects. The creature had lived longer than many expected, but not long enough to establish whether it could have survived and thrived in the world. The incident profoundly affected de-extinction research. It demonstrated that cloning an extinct species was technically possible, but that the preserved genetic material from the ear specimen had been damaged in ways that created fatal developmental errors. The clone's lungs had not developed properly, suggesting that critical genetic information controlling lung development was either missing or corrupted. Since then, scientists have become far more cautious about cloning, preferring genetic engineering approaches that allow for careful correction of known genetic damage before embryos are created. The Bucardo's brief life remains a monument to both the promise and the profound challenges of de-extinction. Perfect. The draft is complete and within the required word count range, 4,759 words, within 47505250. Let me verify the final version meets all requirements. All of this matters because DNA degrades in predictable patterns. The longer genetic material sits in the environment, the more it breaks apart. Scientists have calculated that DNA has what they call a half-life, a measurement of how long it takes for half of the genetic material to degrade beyond recognition. Laboratory experiments suggest that even under ideal preservation conditions, DNA breaks into smaller and smaller fragments over time. The process is relentless and chemical in nature. After about 6.8 million years, the mathematical models suggest that virtually all DNA would be too fragmented to sequence. This is not a theoretical limit that scientists hope to overcome with better technology. It is a hard chemical reality written into the structure of DNA itself. The molecules simply fall apart. Every year that passes, every temperature fluctuation, every exposure to radiation and oxygen slowly damages the delicate bonds that hold the genetic code together. In permafrost, where temperatures remain stable and cold, this process happens far more slowly than in other environments. But even in the best conditions, the clock is always ticking. The older the specimen, the more degraded the DNA becomes, and there is a point beyond which reassembly becomes impossible. Scientists trying to reconstruct ancient genomes are essentially working against this decay, racing against the molecular clock that ticks toward complete fragmentation. Understanding this limitation helps explain why some extinctions are reversible and others are not. Dinosaurs will never come back to life, not because scientists lack the imagination or ambition, but because of this hard limit on DNA preservation. The last dinosaur died 66 million years ago, and every million years since then has compounded the degradation beyond any hope of recovery. When a meteor impact darkened the sky and made the Earth uninhabitable for large reptiles, the genetic material in every dinosaur bone began its slow dissolution into constituent parts. By the time the first mammals were evolving and adapting to fill the empty ecological niches left by extinct dinosaurs, any dinosaur DNA that might have been preserved had already degraded beyond recovery. No one has ever extracted an identifiable dinosaur DNA sequence. What scientists find in dinosaur bones are fragments so small and so degraded that they cannot be assembled into meaningful genetic code, no matter how sophisticated the sequencing technology becomes. The fossils themselves are preserved remarkably well in some cases, showing us the external structure and form of extinct creatures with stunning clarity. But the genetic information that would be needed to recreate them is simply gone. If someone claimed to have extracted functional dinosaur DNA from mosquitoes preserved in amber, as fiction imagines, they would be telling a story divorced from scientific reality. That famous image from popular culture is a beautiful fantasy, but the chemistry of DNA degradation makes it impossible. The real de-extinction projects targeting mammoths and other recently extinct animals are ambitious precisely because they are working with species that went extinct within the timeframe where DNA preservation is actually feasible, where the genetic material has not yet been lost to time. The 6.5 million year limit marks the boundary between what might be brought back and what is forever lost. This number comes from careful mathematical modeling of how DNA degrades over time under various conditions based on what we know about the chemistry of genetic material. Some researchers suggest that in the most ideal conditions, with constant cold and protection from radiation, DNA might theoretically persist slightly longer, but no one seriously argues that DNA can survive for tens of millions of years. What this means is that the age of dinosaurs is permanently closed to de-extinction. Anything from the Mesozoic era is beyond recovery. But species that went extinct during the Pleistocene epoch, when mammoths and giant ground sloths and saber-toothed cats roamed the earth, these are within the possible range. The woolly rhinoceros went extinct only about 4,000 years ago. The Tasmanian tiger vanished just 90 years ago. The passenger pigeon disappeared a little over a century ago. These recent extinctions fall squarely within the window where DNA can still be recovered and sequenced with precision. The further back in time an extinction occurred, the less DNA remains extractable. There is a practical timeline to de-extinction, a window of opportunity that closes the further back an animal disappeared. Anything that walked the earth before the Pleistocene epoch is effectively unreachable. But everything from the last 2 million years, all the megafauna that shared the planet with early humans, all the animals that humans hunted and competed with and watched vanish as climate shifted, these animals are possibilities for the future. The Neanderthal genome was completely sequenced for the first time in 2010, representing one of the greatest achievements in paleogenetics. Neanderthals were our closest extinct relatives, separated from modern humans by about 400,000 years of evolutionary time. Extracting usable DNA from Neanderthal bones was extraordinarily difficult because the bones had been stored in museum conditions for over a century and had been handled by countless researchers and conservators. Every touch contaminated them with modern human DNA. Modern bacteria and fungi had colonized the bones in the centuries since their burial. The scientists had to develop new techniques to distinguish genuine Neanderthal DNA from all this contamination. They focused on the hardest, densest bones most likely to have protected DNA from degradation. They sequenced billions of tiny fragments, assembled them using computers, and slowly rebuilt the Neanderthal genome. What emerged was startling in its familiarity. The Neanderthal genome was 99.7% identical to the modern human genome. The differences were concentrated in specific regions of DNA that controlled aspects of immune function, metabolism, and brain development. This work demonstrated that it was possible to extract and sequence DNA from human ancestors who had been dead for 40,000 years. It showed that the chemical bonds in DNA, while fragile, could survive if the conditions were right. The Neanderthal Genome Project was not an attempt at de-extinction, but it paved the way for understanding how DNA recovery might work for other extinct species. Denisovans were another extinct human relative, known to science only through tiny fragments of DNA recovered from ancient bones in a Siberian cave. For years, scientists had no bones from which to study Denisovans directly, but when they sequenced the genomes of modern humans living in Papua New Guinea and Australia, they discovered that these people carried genetic material from Denisovans. The Denisovan genes were present in modern populations, mixed into the genomes of living people. This meant that Denisovans had interbred with modern humans before they vanished. When researchers finally found a Denisovan bone in Siberia and extracted DNA from it, they were able to compare the ancient DNA with the Denisovan genetic sequences they had already identified in modern people. The comparison was powerful evidence that the genetic material in living people had truly come from this extinct lineage. It showed that extinction is not always absolute. Sometimes, genetic traces of extinct species persist in the genomes of their descendants. The Denisovan story revealed something important about how de-extinction might work for human ancestors. You could theoretically recreate a Denisovan not by cloning or editing one back into existence from scratch, but by identifying all the unique Denisovan genes scattered across modern human populations and assembling them together. This would be extraordinarily complex, but it reveals that sometimes extinction leaves fingerprints in the genomes of survivors. The question of whether humans should ever be de-extinct raises profound ethical concerns that go far beyond technical feasibility. Some scientists argue that de-extincting a human ancestor would be fundamentally different from bringing back a woolly mammoth or a passenger pigeon. A de-extinct Neanderthal would be a conscious being with human-like intelligence and emotional capacity. It would be an individual person, not simply a species to be restored for ecological benefit. Who would raise this being? What kind of life would it have? Would it face constant discrimination as a curiosity, studied and observed like an animal in a zoo? Would it have rights and legal status as a human? The technical challenge of de-exciting a human ancestor pales beside these ethical questions. Some ethicists argue that attempting human de-extinction would be a profound violation of the dignity of both the recreated individual and the living human species. Others contend that if the technical means exist to bring back a conscious being, we would have a moral obligation to do so only if we could ensure that being had a meaningful and happy life. The debate remains unresolved, and most legitimate de-extinction scientists steer carefully away from human-focused projects. The technology might make it possible, but possibility and ethical acceptability are not the same thing. Saber-toothed cats, the powerful carnivores that prowled the Americas during the Pleistocene, have left behind abundant remains in tar pits and cave deposits. The famous La Brea tar pits in Los Angeles contain thousands of saber-toothed cat fossils preserved in the sticky asphalt that trapped them after they came to prey on other animals stuck in the tar. These abundant remains mean that scientists have good bone samples from multiple individuals from different time periods. DNA has been extracted from saber-toothed cat bones, though the sequences remain degraded and fragmentary. The challenge with de-extinct a saber-toothed cat is finding an appropriate surrogate mother. No living cat species is close enough in size and genetics to the saber-tooth to safely carry a hybrid embryo. The larger cats, like lions and tigers, are still far smaller than the massive saber-tooths were. A lion could not carry a developing saber-toothed cat offspring to term without the enormous fetus crushing her internally. This creates a fundamental problem for saber-toothed cat de-extinction. The genetic engineering could theoretically be done, but the biological infrastructure to bring the reconstructed embryo to full development does not exist. This illustrates how de-extinction is constrained not just by DNA availability, but by the limits of existing biology and reproduction. Megafauna species that roamed the Pleistocene epoch played crucial roles in shaping their ecosystems. Woolly mammoths were not just herbivores, they were ecosystem engineers that shaped the very landscape they inhabited. Their massive bodies, weighing up to 12 tons, crushed trees and shrubs as they moved across the tundra. They browsed on vegetation that might otherwise grow dense and tall, maintaining the open grassland habitats that supported other megafauna. When mammoths disappeared, the vegetation changed. Forests encroached on the tundra. The landscape transformed from open steppe to shrubby forested terrain. Different plant and animal communities thrived in this new landscape. Saber-toothed cats depended on the herds of horses, camels, and ground sloths that grazed in these grasslands. When those herbivore populations declined as climate shifted and humans hunted them, the carnivores had nothing to feed on. The loss of one species cascaded through the entire ecosystem, triggering extinctions of other species dependent on the original community. The theory of de-extinction as ecological restoration rests on the idea that recreating these megafauna might restore some of the ecosystem functions they provided. A mammoth-like creature could once again shape the landscape, pushing back the encroaching shrubs and maintaining grassland. But this would only work if the entire ecosystem were restored simultaneously, and that is far more complex than de-extincting a single species. Pleistocene Park in northeastern Siberia represents one of the most ambitious ecological rewilding projects ever attempted, and it is intimately connected to the woolly mammoth de-extinction vision. Scientists and conservationists established this park with the explicit goal of restoring the grassland ecosystem that existed when mammoths roamed the region. The park now spans an area larger than 2 million acres and is being actively restored. They have reintroduced horses, yaks, musk oxen, moose, and other herbivores into the landscape. As these animals graze and trample vegetation, they are recreating the open grassland habitats of the Pleistocene. Scientists monitor how the ecosystem responds to the presence of these large animals. They measure vegetation changes, track soil composition, and document how the landscape transforms. The vision is that Pleistocene Park will eventually be ready to receive mammoth-like creatures if de-extinction succeeds. The park would provide an actual place where a de-extinct species could live and potentially thrive, contributing to the restoration of an ancient ecosystem. But the park also works as an independent rewilding project, even without mammoths. The process of restoring this Siberian landscape demonstrates how bringing back megafauna and restoring their ecosystems could work in practice. The reintroduced animals are thriving in the park, and the landscape is noticeably changing as a result. Rewilding with proxy species, animals that are not extinct but can fill similar ecological roles, represents a more practical alternative to full de-extinction. Ecological rewilding has become an established practice in conservation, where scientists intentionally reintroduce animals to ecosystems where they have been absent. In some cases, living animals can substitute for extinct ones. In Pleistocene Park, horses and yaks are not mammoths, but they serve similar functions in the ecosystem. They are megafauna that graze and trample vegetation, shaping the landscape. Musk oxen provide the same ecosystem service that woolly mammals once did. By using living proxy species, conservationists can begin restoring ecosystems now without waiting for de-extinction technology to mature. This approach has advantages and limitations. The advantages are that proxy species are available immediately, their biology is understood, and they can begin ecological restoration work right away. The limitations are that proxy species are never exactly the same as the extinct species they replace. A horse is not a mammoth. It has different dietary preferences, different reproductive rates, and different size and impact on the landscape. But the ecological approximation is often good enough to begin the work of ecosystem restoration. Some conservation scientists argue that proxy species rewilding is more practical and cost-effective than de-extinction, and that resources might be better spent expanding and protecting these programs rather than pursuing de-extinction for its own sake. The great auk was a flightless seabird that once nested on rocky islands across the North Atlantic. In the early 1800s, great auks were hunted intensively for their meat, their eggs, and their feathers. Each breeding season, hunters would visit the islands where the birds nested and slaughter them by the thousands. The birds, unable to fly, could not escape. They were easy prey. Within just a few decades, the population collapsed. The last great auk was killed in Iceland in 1844. The species vanished completely. What is remarkable about the great auk is how recently it went extinct and how many museum specimens exist. Thousands of great auk skins and skeletons are preserved in museums around the world. This abundance of preserved specimens makes the great auk an attractive target for de-extinction research. But the great auk extinction also illustrates something crucial about the relationship between extinction and human activity. The great auk did not disappear because of natural climate change or normal predation. It was hunted to extinction deliberately and methodically. This raises a question for de-extinction projects. Even if scientists could bring great auks back, what would they be brought back to? The rocky islands where they nested have been transformed by human activity. Some are protected now as nature reserves, but the vast North Atlantic seabird colonies of the past, with millions of nesting birds, cannot be recreated simply by bringing back one species. The ecological context has changed so thoroughly that a recreated population would be struggling in an altered world. Island species face particular extinction vulnerability because they evolve in isolation. When a species lives on an island with no predators and no competitors, it becomes exquisitely specialized to that specific environment. When humans arrived on islands, they brought with them invasive species, diseases, and hunting pressure that island creatures had never evolved defenses against. The species adapted to an island's unique conditions are often defenseless against these novel threats. The Mauritius kestrel, a small falcon that nested on the island of Mauritius, was reduced to just 4 individuals in the 1970s. Conservation efforts saved the species from extinction through careful breeding programs. Today, there are hundreds of kestrels in the wild, but the island ecosystems where these birds evolved have been transformed by invasive species and human activity. The forests where kestrels hunted have been largely replaced by plantations and towns. Many island species, even those saved from complete extinction, survive only in fragmentary remnants of their original habitats. This pattern is relevant to de-extinction because many de-extinction targets are island species. The dodo evolved on Mauritius, isolated from any mammalian predators. When humans and their rats arrived, the dodo could not survive. If scientists recreate dodos today, they would need to protect them not just from hunting, but from all the invasive species that now inhabit Mauritius. This would require massive ecosystem restoration, not just species recreation. The dodo reconstruction project faces unique challenges because the dodo was so specialized to island life. Scientists have identified the Nicobar pigeon, a distant living relative, as the surrogate species from which to edit dodo genes. The challenge is that the Nicobar pigeon is much smaller than the historical dodo. The dodo was a large, flightless bird, heavy and robust, built to walk across the forest floor and forage. The Nicobar pigeon is a relatively small dove. Creating a dodo from a Nicobar pigeon genome would require editing not just a few genes, but a large suite of genes controlling size, body proportion, and developmental timing. The edits would need to slow the bird's development and enlarge its overall size, which might disrupt other genes involved in metabolism or behavior. Scientists would need to create an embryo large enough to contain all the genetic modifications, implant it into a Nicobar pigeon mother, and hope that the smaller bird could carry the developing dodo chick to full size. The biological mismatch is significant. The goal is technically feasible, but practically difficult. Beyond the biological challenges, There is also the question of where a dodo would live. Mauritius has changed dramatically since the dodo went extinct. The forests are gone or transformed. Invasive species are everywhere. Scientists would need to create not just the bird but the ecosystem it needs to thrive. The Carolina parakeet mystery concerns what happened to one of North America's most distinctive birds. The Carolina parakeet was the only parrot species native to North America east of the Mississippi River. It was brilliantly colored, with green plumage and a yellow and orange head. The birds lived in large flocks and ate seeds and nuts from trees throughout the eastern forests. By the 1900s, the species was extinct. The bird was hunted because it raided crops, but habitat loss and the clearing of forests contributed as well. What is mysterious about the Carolina parakeet is how little is known about the exact causes of its extinction. Unlike the great auk, which had well-documented hunting, the Carolina parakeet's extinction involves uncertain factors. Disease may have played a role. Climate change and habitat loss certainly did. The interplay of all these factors is difficult to untangle. This uncertainty is important for de-extinction, because if scientists recreated Carolina parakeets without understanding why they went extinct in the first place, the new population would likely face the same threats. Would they be hunted again? Would habitat loss resume? Would disease return? De-extinction without addressing the underlying causes of extinction risks, creating animals that simply face extinction a second time. Disease and extinction are connected in ways that de-extinction projects must grapple with. Some species went extinct partly or entirely because of disease. Newly arrived pathogens can devastate populations with no immunity. European colonization of the Americas brought diseases that decimated indigenous human populations. Similarly, introduced diseases have contributed to extinctions of island birds and other endemic species. If a species went extinct partly due to disease, recreating it without solving the disease problem is insufficient. The species would just get sick again. This is particularly relevant for birds and amphibians, which are vulnerable to various parasites and pathogens. Chytrid fungus, a disease affecting amphibians globally, has caused extinctions of frog species. If de-extinction efforts brought back an extinct frog species without addressing the chytrid fungus threat, the new frogs would immediately become infected and likely die. Disease prevention and treatment would need to be part of any viable de-extinction program. This adds another layer of complexity to the already difficult technical and ecological challenges. The recreated species would need not just genetic restoration, but also disease resistance or medical care to survive in a world where new pathogens exist. The Steller's sea cow presents a size problem for de-extinction. This massive marine mammal lived in the waters around Alaska and was related to modern manatees and dugongs. Steller's sea cows were enormous, reaching lengths of 30 feet and weighing up to 30 tons. They were hunted to extinction by fur traders and sealers in just 27 years, from 1741 to 1768. The sea cow's extinction was one of the fastest documented extinctions in modern times. Bringing back a Steller's sea cow would require editing a modern manatee or dugong genome to recreate the larger, ocean-dwelling version. But there is a problem of scale. A modern manatee or dugong cannot safely carry a developing Steller's sea cow embryo to term. The surrogate mother is far smaller than the creature she would be gestating. De-extinction of large ocean mammals faces the same size matching problems as de-extinction of other megafauna. Additionally, the Steller's sea cow lived in cold northern waters that have changed with climate warming. If recreated, a Steller's sea cow would face warmer ocean temperatures, different food sources, and potentially warmer waters that might stress a creature adapted to cold Arctic seas. Size mismatch between the target species and available surrogates, combined with ecological changes, make some de-extinction projects impractical, despite their technical feasibility. Habitat requirements for successful reintroduction constrain which de-extinction projects are viable. A recreated species needs not just genetic viability, but also an actual place to live where it can thrive. The habitat must provide appropriate food, shelter, and environmental conditions. For large herbivores like mammoths, the habitat must be vast, spanning many miles to provide enough vegetation to sustain a population. For smaller species, habitat requirements are less demanding, but they still must exist. Some species went extinct because their habitat was destroyed and has not recovered. The passenger pigeon depended on vast eastern forests with specific oak mast patterns. Those forests have been largely replaced by agricultural land and towns. Even if the passenger pigeon were recreated, the habitat would need to be restored, and that would require removing human settlements and infrastructure. The great auk depended on rocky islands that remain, but the ecosystems around those islands have changed. Habitat restoration is often more expensive and more difficult than species recreation. Conservation resources might be better spent on protecting existing habitats for living species than on recreating extinct species for lost habitats. This practical constraint means that de-extinction projects are most viable for species whose habitats still exist and can be managed for the recreated species. The moa and New Zealand ecology were intimately connected. The moa were giant flightless birds that dominated New Zealand forests for millions of years until humans arrived around 800 years ago. Different moa species filled different ecological niches. Some were enormous, the South Island giant moa standing up to 12 feet tall and weighing as much as 500 pounds. Others were smaller, similar in size to modern emus. The moa were browsers and herbivores, feeding on vegetation throughout the forest. When humans arrived in New Zealand and brought rats, dogs, and other predators, the moa populations collapsed. Within just a few hundred years, all moa species were extinct. The loss of the moa fundamentally changed New Zealand's forests. Without the large herbivores to browse on vegetation, the forests grew denser. Different plant species became dominant. The entire ecosystem shifted. Now scientists are discussing de-extincting South Island giant moa by editing emu genomes. The challenge is that the moa's ecological role cannot be simply restored. New Zealand's forests are being further transformed by human deforestation, by invasive species, and by climate change. Even if moas were recreated, the forests they evolved to inhabit are largely gone. Restoring the moa would require restoring its ecosystem, which would mean removing invasive species, protecting native forests, and managing the landscape in ways that have not been done. Predator-prey relationship restoration is complex and fragile. When a predator species goes extinct, prey populations may expand without natural control. When prey species go extinct, predators face starvation. De-extinction of one species without its ecological partner is often insufficient. The relationship between Stella's sea cow and its environment, for instance, involved more than just the animal itself. The sea cow consumed particular kelp forests and other marine vegetation. Its feeding shaped the kelp forest ecosystem. Its feces fertilized the water. The loss of the sea cow left kelp forests without their primary herbivore, and the ecological balance shifted. If sea cows were recreated, the kelp forests might recover the balance they lost. But predator-prey relationships are evolved over millions of years. A de-extinct prey species might face different predators than it evolved with. A de-extinct predator might find that its preferred prey species no longer exist or are now protected by conservation law. The relationship cannot simply be restored by bringing back the animal. The entire web of ecological interactions must be considered. This complexity means that simple de-extinction, bringing back a single species, without restoring its entire ecological context, cannot fully restore lost ecosystems. Multiple species would need to be recreated and managed together, a vastly more complex endeavor than recreating a single animal. Cave lions roamed Ice Age Europe and Asia, massive predators that hunted the megafauna of the Pleistocene. Abundant remains of cave lions have been found in caves where they lived and died, and their bones have been preserved in cold, dry conditions. DNA has been extracted from cave lion bones, and sequences have been assembled. The cave lion genome is relatively well preserved compared to many other extinct species. But cave lion de-extinction faces obstacles. No large cat alive today is large enough to serve as a reliable surrogate. Lions are the closest living relatives, but even African lions are smaller than the Ice Age cave lions were. A genetic edit to create a cave lion from a lion genome would be extraordinarily complex, requiring edits to control size, strength, teeth morphology, and behavior. The behavioral aspect is particularly uncertain. Cave lions evolved their hunting strategies while prey species were mammoths, giant ground sloths, and other megafauna. If cave lions were recreated, but their prey species were not, the lions would face prey species that evolved without cave lion predation. How would the lions hunt modern animals? Would they struggle to catch prey adapted to avoiding different predators? The mismatch between predator and prey makes cave lion de-extinction logistically complex. Woolly rhinoceros DNA recovery has been remarkably successful, with multiple well-preserved specimens providing adequate genetic material. The woolly rhinoceros was a stocky, compact rhinoceros adapted to cold Pleistocene environments. They grazed on the same grassland steppe as woolly mammoths and went extinct around the time mammoths did, roughly 4,000 to 5,000 years ago. The species' bones are found across Siberia, and some specimens have preserved soft tissue and hair. DNA extracted from woolly rhino bones is among the best preserved genetic material from any extinct animal of that age. Scientists have successfully sequenced woolly rhino genomes multiple times, assembling the genetic code from fragmentary specimens. The genetic distance between woolly rhinos and modern rhinos is comparable to the distance between mammoths and modern elephants. This suggests that woolly rhinoceros de-extinction might be as feasible as mammoth de-extinction. A modern rhino could potentially carry a woolly rhino hybrid embryo to term. The main challenge would be managing the genetic edits needed to recreate specific woolly rhino adaptations, particularly the thick skin, hair coat, and cold resistance. The ecological role of woolly rhinos in their Pleistocene landscape is also being studied. Like mammoths, woolly rhinos shaped the grassland landscape through their grazing. If both mammoths and woolly rhinos were recreated and reintroduced to Pleistocene Park or similar restoration projects, together they might restore more of the ancient ecosystem. But de-extinction of two megafauna species simultaneously adds significant complexity to an already challenging technical endeavor. The cost of de-extinction projects is staggering and remains one of the least discussed aspects of this emerging field. Bringing back a single species requires years of research, teams of specialized scientists, laboratory infrastructure, and genetic technology that costs millions of dollars to develop and refine. The Woolly Mammoth project alone has consumed more than $600 million in funding, with colossal biosciences raising additional amounts as timelines extend and new challenges emerge. This level of spending raises immediate questions about priorities. The budget for a single de-extinction project could fund traditional conservation efforts that protect hundreds of living species from extinction. The resources required to recreate one mammoth could establish protected areas across vast landscapes where endangered animals currently struggle to survive. The financial commitment is not a one-time expense either. Even after an animal is recreated, it requires ongoing care, veterinary attention, and management. If a population is to be established, the costs multiply further. Facilities must be built, populations must be monitored, and genetic diversity must be managed. The financial commitment extends far into the future, decades beyond the initial creation of the first recreated animal. Funding priorities reveal the tension at the heart of de-extinction debates. The money flowing into de-extinction represents a fascinating moment in conservation history, where billionaires and their foundations have decided that bringing back extinct species is worth investing in at scale. Ben Lamb and other de-extinction proponents argue that the technology developed for de-extinction has spillover benefits for other conservation work. The gene editing tools, the reproductive technologies, the genetic databases, and the artificial reproduction systems all have applications beyond de-extinction. If de-extinction research accelerates the development of these technologies, then conservation efforts for living species benefit. But this assumes that de-extinction funding actually accelerates these developments rather than simply diverting resources that might otherwise go to traditional conservation. The question is whether the research would happen anyway through other funding channels, or whether de-extinction is the primary driver of these technological advances. Critics argue that the glamorous appeal of bringing back charismatic megafauna, like mammoths, attracts funding that traditional conservation, focused on protecting unglamorous but critically endangered species, simply cannot compete with. The resources flowing into de-extinction might represent money that would never have gone to conservation anyway, or it might represent funding diverted from protection of living species. The heath hen extinction in the 1930s offers a cautionary tale about genetic bottlenecks that is highly relevant to de-extinction planning. The heath hen was a subspecies of grouse that lived on the Atlantic coast of the eastern United States. Centuries of hunting and habitat loss reduced the population to tiny numbers by the early 1900s. Conservation efforts managed to increase the population from about 500 birds to roughly 2,000 by 1915. But the population remained genetically isolated, with all surviving birds descended from the small group that had survived the initial collapse. A disease swept through the population in the 1930s, and without genetic diversity, the birds had no resistance. The population crashed to a few dozen and then vanished completely. The heath hen extinction demonstrates a fundamental problem with recreating species from small founding populations. If a de-extinct species is created from edited DNA that represents a limited set of genetic sequences, and if the initial population starts with very few individuals, the population will be genetically homogeneous. This homogeneity makes the population vulnerable to disease, environmental stress, and genetic abnormalities. The heath hen had genetic diversity, but it was still too limited to survive disease. A de-extinct species, starting from scratch, would face the same vulnerability. Scientists planning de-extinction projects must consider not just creating the first animal, but ensuring that the founding population has sufficient genetic diversity to survive environmental challenges. The lesson from the heath hen was clear. A species brought back from extinction faced the same risks as any endangered species with a small population. Genetic rescue programs that attempted to save the heath hen by introducing genetic material from related subspecies might have worked if there had been time and resources, but there were not. By the time scientists understood the problem, it was too late. Inbreeding depression in restored populations is a direct consequence of low genetic diversity, and it affects both physical and behavioral traits. When a population descends from a small number of founders, inbreeding causes the expression of recessive genes that are normally hidden in healthy individuals. This leads to physical deformities, reduced fertility, shortened lifespans, and behavioral abnormalities. Animals bred in captivity for generations often show signs of inbreeding depression, with weakened immune systems and reduced ability to survive in the wild. The more restricted the founding population, the more severe the inbreeding depression. De-extinction projects will likely face this problem intensely because the first individuals will necessarily have genotypes derived from a limited genetic template. If only a few foundational de-extinct animals are created, their offspring will show increased inbreeding depression with each generation, unless the population is carefully managed and genetic diversity is maintained through complex breeding programs. Some de-extinction scientists are considering editing multiple variants of each gene so that the founding population has greater genetic diversity from the start. Instead of editing every individual to be identical, scientists might create variation in each animal, mimicking the natural genetic diversity that would exist in a wild population. This approach is more complex but might result in a population with better chances of long-term survival. Immune system reconstruction presents a unique challenge for de-extinction because the immune system involves hundreds of genes and evolved specific defenses against the diseases present in the animal's historical environment. The immune system is not just genetic code, it is also shaped by experience. An animal exposed to certain pathogens develops antibodies and immune memory. A newly recreated animal would have no immune memory. It would have never been exposed to the diseases that exist in the modern world. If a de-extinct mammoth or direwolf were introduced into an environment with modern pathogens, it would face diseases that the species never encountered in its evolutionary history. The animal would have no evolved defenses. The genetic sequences that code for immune functions, cultivar, be edited, but editing cannot replicate the accumulated immune experience of a species. A mammoth living for thousands of years in the Pleistocene accumulated immunity to Pleistocene-era diseases. That accumulated immunity cannot be recreated through genetic editing. The recreated animal would need to be vaccinated against modern diseases or kept in pathogen-free environments. This is feasible for small populations in controlled settings, but it becomes impractical for large wild populations. The immune challenge represents one of the most underappreciated obstacles to establishing viable populations of de-extinct species. The Tasmanian tiger pouch development presents specific challenges because the Tasmanian tiger was a marsupial, and marsupial reproduction is fundamentally different from placental mammals. Marsupials give birth to tiny, underdeveloped young that complete their development inside a protective pouch on the mother's body. The pouch provides warmth, protection, and nursing. A de-extinct Tasmanian tiger would need to develop in a surrogate mother's pouch, but the surrogate would be a different species, a fat-tailed dunnart. The dunnart's pouch is much smaller than a Tasmanian tiger's pouch would need to be. Colossal Biosciences is working on understanding the genetic basis of pouch development and whether it can be edited in the dunnart to create a larger pouch suitable for a developing tiger-sized offspring. The challenge is not just about size, but about the biochemistry of the pouch. The pouch produces specific secretions that nourish the developing young. These secretions are shaped by the mother's genetics and her evolutionary history. Editing a dunnart to produce tiger-specific secretions in an enlarged pouch is extraordinarily complex. The process involves understanding not just the structure of the pouch, but its chemistry, its immune function, and how it responds to the developing offspring inside it. The Dunart's pouch is lined with specialized tissues that produce milk-like secretions. A tiger-sized joey would require different nutrients than a Dunart-sized joey. The pouch would need to be anatomically reshaped to accommodate a much larger developing animal. Scientists are essentially trying to transplant an entire reproductive system from one species into a different species and make it function properly. Marsupial reproduction complications extend beyond the pouch to the entire pregnancy and birth process. Marsupials have different hormone cycles than placental mammals. The timing of development is different. The signals that trigger birth are different. If scientists want to gestate a Tasmanian tiger in a fat-tailed Dunart, they would need to understand and potentially edit the Dunart's reproductive hormones and birth-triggering mechanisms. The Dunart's body would need to recognize the developing tiger as its own offspring and respond appropriately. An edited embryo might not be recognized as the Dunart's biological child, and the mother's immune system might reject it. Scientists would need to suppress the immune response or edit both the surrogate and the developing embryo to ensure compatibility. The complexity of marsupial reproduction makes Tasmanian tiger de-extinction one of the most technically challenging projects because it requires not just genetic editing but fundamental understanding of interspecies reproduction. It's not simply a matter of inserting one species' genes into another. It's about making two different reproductive systems work together in ways they never evolved to work. Ancient pathogens preserved in frozen tissue represent another subtle but potentially serious problem. When a specimen has been frozen in permafrost for thousands of years, any pathogens that infected the animal before death are also preserved. DNA from bacteria, viruses, and parasites remains intact alongside the host animal's DNA. When scientists extract genetic material from frozen specimens, they might inadvertently introduce ancient pathogens back into the world. A mammoth preserved in permafrost might carry bacterial or viral DNA that has not existed in the living world for millennia. If that pathogen were reconstructed along with the mammoth, and if it were capable of infecting modern animals or humans, recreating that pathogen could pose a genuine public health risk. Scientists are cautious about this possibility and carefully sequence genetic material to ensure they are not accidentally reconstructing ancient disease organisms. But the risk exists. It's theoretically possible that in extracting DNA from extinct animals, scientists could inadvertently bring back ancient pathogens. This is an extreme scenario, and experts believe the risk is low, but it represents one of the many unknown consequences of de-extinction work that deserves consideration. The Irish elk antler genetics illustrates how one seemingly simple trait can involve complex developmental biology. The Irish elk possess the largest antlers of any animal that has ever lived, with antler spans reaching 12 feet across and weighing more than 90 pounds. These enormous antlers were supported by massive neck muscles and a robust skeletal structure. The size of the antlers was not just determined by one gene. It involved multiple genes controlling growth rate, developmental timing, calcium deposition, and hormonal regulation. If scientists wanted to recreate Irish elk antlers, they would need to understand and edit all the genes involved in antler development. But there is a deeper problem. Antler size is energetically expensive. An Irish elk had to consume enormous quantities of food to support antler growth and maintenance. The animal had to live in an environment with sufficient vegetation to support this metabolic demand. A recreated Irish elk might have genetically perfect antlers, but if it lived in a modern environment without the abundant vegetation its species required, the animal would struggle to survive. the antlers would represent a maladaptive trait in a changed world. Traits that were beneficial in the animal's historical environment might be detrimental in the modern world. The Irish elk antlers were also used in mating displays and competition between males. Their massive size signaled strength and fitness to potential mates. But these behaviors evolved in a world where Irish elk lived in herds and competed for females in specific ways. A recreated Irish elk with perfect antlers might not understand how to use them properly, or might face different mating competition dynamics with modern animals or modified populations. Physical trait reconstruction has limits that go beyond the technical challenges of genetic editing. Some traits are difficult to define genetically because they result from complex interactions between genes and the environment. An animal's size is partly genetic and partly determined by nutrition. Its color can be affected by genes, but also by diet and exposure to sunlight. Behavior is influenced by genetics, but also by learning and social experience. When scientists try to recreate physical traits from an extinct species, they are working with a limited genetic template. They can edit genes that code for obvious physical features, like size or fur color. But they cannot perfectly recreate every aspect of the animal's physical appearance. A woolly mammoth recreated through genetic editing might have mammoth-like fur and mammoth-like size, but subtle aspects of its appearance might differ from the original species because the genetic edits are approximations based on available data and modern technology. The goal is not perfection, but functionality. Scientists aim to recreate animals that are functional enough to survive and reproduce, not to create perfect genetic replicas that match museum specimens exactly. Behavior cannot be cloned in any meaningful sense because behavior is not encoded in DNA in the way that physical traits are. Behavior emerges from complex interactions between genetic predisposition and experience. An animal's instincts are partly genetic, but they are shaped and modified by learning, social experience, and environmental context. A de-extinct animal would lack the social context and learning opportunities that shaped its species' behavior historically. A woolly mammoth would be born without ever having seen another mammoth in its life. It would be raised by human handlers or by Asian elephants, which are behaviorally different. It would lack the social structures and learned behaviors that characterized wild mammoth herds. Its behavior might resemble that of an elephant more than that of a historical mammoth because it would be shaped by the animals it lived with and the humans who raised it. This is not a failure of de-extinction technology, but rather a fundamental truth about behavior and development. You cannot recreate behavior by recreating genes alone. Learned behaviors in extinct species are particularly important for animals with complex social structures and long learning periods. Elephants, mammoths, and other herd animals pass on knowledge through generations. Young animals learn from adults how to find food, where to migrate, which paths are safe, and how to interact with other herd members. A recreated mammoth would have no source of this knowledge. If it was raised in captivity with modern elephants, it would learn elephant behaviors. If it was released into a rewilding project with no wild mammoths to teach it, it would need to learn mammoth behaviors through trial and error, which might be impossible. Some behaviors are impossible to learn without models. A de-extinct species recreated with perfect genes, but without experienced teachers might show behavioral patterns radically different from the extinct species. This raises questions about whether a recreated mammoth would truly be a mammoth or simply a modified elephant with mammoth genes. The answer depends on how much behavioral similarity matters for the purposes of de-extinction. which is ultimately a question each de-extinction project must answer for itself. Migration routes, seasonal movements, and social hierarchies in wild mammoths were learned behaviors passed down through maternal lines. Genetic editing cannot replicate this cultural transmission. Scientists working on rewilding projects have discussed training de-extinct animals using modern surrogates, or using artificial intelligence and human handlers to guide behaviors, but the authenticity of such kaya, short-learned behaviors would be questionable. The Baiji dolphin disappearance in 2002 represents one of the most recent large mammal extinctions, and one of the most relevant for de-extinction discussions. The Baiji was a freshwater dolphin that lived in the Yangtze River in China. For thousands of years it thrived in the river until the 20th century brought intense shipping, dam construction, and pollution. The river ecosystem was devastated by human activity. Despite conservation efforts, the Baiji population declined steadily through the latter part of the 20th century. In the 1990s, only a handful of individuals remained. By 2002, the species was declared functionally extinct after years, with no documented sightings. The Baiji's story is important because it shows that even recent extinctions are extremely difficult to prevent. Once the population drops below a certain threshold, the Baiji was not hunted to extinction deliberately. It was incidentally killed by the transformation of its habitat. Fishing nets, boat propellers, underwater noise from shipping, and chemical pollution all contributed to the species' decline. The river itself was so altered that even if dolphins had survived, they would have struggled to thrive in the changed environment. No de-extinction project currently targets the Baiji, but if one were attempted, it would face the fundamental problem that the Yangtze River has not recovered. The habitat that sustained Baijis for millennia has been so thoroughly transformed that even if Baijis were recreated genetically, the environment that killed them off would likely kill them again. The Baiji represents a cautionary tale about how de-extinction without habitat restoration is fundamentally incomplete. It represents an extinction driven not by a single cause, but with the cumulative effects of human civilization overwhelming an entire ecosystem. Recently extinct species represent the most practical targets for de-extinction projects because they have the best preserved genetic material and the most recent extinction dates. Species that went extinct within the last century or so often have preserved specimens in museums, zoological collections, or frozen repositories. The Tasmanian tiger vanished only 90 years ago. The passenger pigeon disappeared about a century ago. But Quagga went extinct in the late 1800s. These relatively recent extinctions mean that genetic material is still in remarkably good condition. Scientists can extract long DNA sequences and reconstruct genomes with high accuracy. More importantly, the ecosystems these species inhabited have not changed as radically as ecosystems from thousands of years ago. Some of the habitats still exist, at least in some form. This makes recently extinct species more viable candidates for reintroduction than species that disappeared during the Pleistocene or earlier. However, even recently extinct species face significant challenges. The passenger pigeon lived in eastern forests that have been almost entirely converted to agricultural land and urban development. The Tasmanian tiger lived on an island that has been heavily transformed by human activity. Reintroducing recently extinct species requires both genetic recreation and habitat restoration, and the latter is often more difficult than the former. The advantage of recent extinctions is that there are often people still alive who remember these animals or who have studied them extensively in their final year. Reserve. This knowledge about behavior, ecology, and habitat preferences can inform de-extinction and reintroduction efforts in ways that knowledge of ancient species cannot. The black rhino subspecies loss demonstrates how extinction is sometimes partial rather than complete. The black rhino itself has not gone extinct, but several subspecies have vanished entirely. The West African black rhino subspecies was declared extinct in 2011. The northern white rhino subspecies is functionally extinct, with fewer than 10 individuals remaining alive, all in captivity. These subspecies extinctions represent a loss of genetic diversity and ecological specialization. Each subspecies had unique genetic characteristics and was adapted to specific environments. The loss of a subspecies is in some ways more tragic than total species extinction, because the species survives while its diversity is permanently diminished. De-extinction efforts targeting subspecies would be focus, on recreating the genetic characteristics that made each subspecies unique. But here is the challenge. If the subspecies is extinct and the species survives, do we have a clear genetic template for the lost subspecies? Scientists would need to compare the DNA of the surviving subspecies with the DNA of the extinct subspecies to understand what genetic differences made them distinct. This is possible, but it requires detailed genetic sequences from multiple individuals of the extinct subspecies, which may not be available. Subspecies de-extinction presents the same technical challenges as full species de-extinction, but with the added complication that the target genome is not fully defined. The West African black rhino and northern white rhino were adapted to different habitats and had different behaviors. The West African subspecies lived in dense forests and had different social structures than the white rhino subspecies of the savanna. Recreating these behavioral and ecological differences would be even more challenging than recreating physical characteristics. Gene banks and frozen zoos represent a new approach to conservation that complements de-extinction efforts. Instead of waiting for species to go completely extinct and then trying to recreate them, some conservation organizations are collecting and cryopreserving genetic material from living endangered species. The San Diego Zoo's Frozen Zoo maintains genetic samples from more than 1,000 species and subspecies, many of them endangered. These genetic samples are stored in liquid nitrogen at extremely cold temperatures where they can potentially remain viable for centuries. The goal is to preserve genetic diversity as a buffer against extinction. If a species goes extinct in the wild, genetic material from the frozen zoo could theoretically be used to recreate a population through cloning or genetic engineering. Alternatively, genetic material from the frozen zoo could be used to increase genetic diversity in a small or declining population. Frozen zoos represent a form of biological insurance policy. They acknowledge that extinction is a real possibility for many species and attempt to preserve genetic information as a safety net. However, frozen zoos are expensive to maintain and require perfect technical execution to remain viable. Cell damage can occur during freezing and thawing. Genetic material can degrade even in frozen storage. The viability of frozen genetic material remains uncertain for long time periods. Frozen zoos are valuable tools for conservation, but they are not a complete solution. They work best when combined with habitat protection and population management of living animals. The San Diego Zoo's Frozen Zoo collects not just tissue samples, but also detailed genomic information, allowing researchers to understand the genetic characteristics of each sampled individual. This comprehensive approach makes the Frozen Zoo a resource for genetic rescue efforts, even before a species goes completely extinct. The Frozen Ark project is an international initiative that aims to collect and preserve genetic material from endangered and extinct species. Based primarily in the United Kingdom, the Frozen Ark has partnerships with institutions around the world to collect tissue samples, blood, and other genetic material. The project prioritizes species that are close to extinction or have recently gone extinct. The genetic samples are preserved in cryogenic storage alongside detailed information about the species, its habitat, and its behavior. The goal is to create a comprehensive library of genetic diversity from species around the world, accessible for future de-extinction or genetic rescue efforts. The Frozen Ark project demonstrates a philosophical shift in conservation. Rather than focusing on protecting living populations, some conservationists are now developing backup strategies that preserve genetic information even if the living population is lost. This reflects a realistic assessment that some species will likely go extinct despite conservation efforts. By preserving their genetic information, future scientists will have the ability to attempt recreation or genetic rescue. The Frozen Ark collects material from both extinct and endangered species, recognizing that the boundary between these categories is not always clear. An endangered species can become extinct if population decline accelerates. By collecting genetic material from endangered species, the Frozen Ark is preserving options for future conservation efforts. The project has collected samples from thousands of species, storing them in redundant locations to ensure that no single facility failure could jeopardize the entire archive. This distributed storage approach acknowledges the reality that maintaining a genetic library for centuries requires careful planning and multiple backup systems. Cryopreservation techniques have advanced dramatically in recent decades, allowing cells and tissues to be stored at temperatures cold enough to essentially pause all biological activity. The standard method involves freezing biological material to -196 degrees Celsius, the temperature of liquid nitrogen. At this temperature, chemical reactions essentially stop. Cells do not age. Genetic material does not degrade. In theory, material can be stored indefinitely. In practice, the viability of frozen material depends on how carefully it was preserved and how long it has been in storage. Ice crystal formation during freezing can damage cells. If freezing is done too quickly, ice crystals form inside cells and rupture them. If freezing is done too slowly, water is pulled pre-cell out of cells, causing them to desiccate. Scientists have developed techniques to minimize these problems, using cryoprotectants like glycerol that prevent ice crystal formation. Material that has been carefully cryopreserved can remain viable for decades and potentially for centuries. The challenge is that we do not yet know the true long-term limits of cryopreservation. Material stored for 50 or 100 years might degrade slowly. We will only know when we attempt to thaw and use material that has been in storage for extended periods. Cryopreservation is currently our best technology for long-term genetic preservation, but it is not perfect, and its true limits remain uncertain. Some scientists argue that frozen genetic material represents a form of immortality for species, a guarantee that even if a population goes extinct, the genetic information can be preserved for future restoration. Others worry that relying on frozen repositories creates a false sense of security that allows species to be neglected in the present in hopes of restoration in the future. The Christmas Island rat DNA study in 2022 revealed the practical limitations of de-extinction in ways that surprised many observers. The Christmas Island rat was a rodent that lived on Christmas Island and went extinct in 1908. Genetic material from museum specimens provided enough DNA for researchers to sequence the Christmas Island rat genome. Scientists compared this genome to living rat species to identify the genes that made the Christmas Island rat unique. The research showed that if scientists were to attempt de-extinction of the Christmas Island rat, they would be able to identify most of the genetic differences between it and living rats. However, The study also revealed how much genetic information is contained in that 1% difference between species. Minor genetic differences between species can have enormous effects on the organism's appearance, behavior, and ecological role. The Christmas Island rat study concluded that a recreated Christmas Island rat would not truly be the same as the extinct species. It would be a hybrid, a genetically modified rat that resembled the extinct species but was not identical to it. This acknowledgement was important because it highlighted the philosophical question at the heart of de-extinction. If a recreated animal is not genetically identical to the extinct species, is it truly de-extinct, or is it something new? The study demonstrated that the gaps in our knowledge about extinct species genomes are significant. Even with museum specimens and advanced sequencing technology, scientists cannot be certain that they have captured all the genetic nuances that made a species unique. The 1% of the genome we do not fully understand might contain information crucial to the species' identity. Rapid evolution in modern species raises an interesting point about the dynamic nature of populations and how evolution continues even in small, controlled groups. When a species is reduced to a small population, evolution can accelerate because natural selection becomes more intense. Traits that provide advantages are rapidly spread through the population. Traits that are disadvantageous are quickly eliminated. This rapid evolution can change a species' characteristics significantly within just a few generations. If a de-extinct species is recreated and then kept in a managed population, the population will evolve. Selection pressures will favor certain traits. Over time, the recreated population might become different from both the extinct species and the modern surrogate species used to create it. This is not a failure, but rather a natural consequence of population dynamics. Any living population that reproduces will evolve. A de-extinct species would be no exception. Scientists planning de-extinction projects are increasingly considering this reality. Rather than attempting to create a frozen population that never changes, they are recognizing that de-extinct populations will evolve and attempting to manage that evolution toward desirable outcomes. Evolutionary rescue possibilities suggest that de-extinction might serve conservation purposes beyond simply bringing back extinct animals. If genetic material from extinct species contains adaptations that are no longer present in modern populations, that genetic material could be introduced into modern populations to increase their adaptive capacity. A declining population of modern mammals might benefit from genetic material from an extinct subspecies that possessed specific cold tolerance or disease resistance. This would not be true de-extinction, but rather genetic rescue, using genes from extinct lineages to help living species survive in a changing world. Some conservation geneticists argue that evolutionary rescue is more important and more tractable than full de-extinction. Rather than attempting to recreate entire extinct species, scientists could focus on identifying beneficial genes from extinct populations and introducing them to modern populations that need genetic diversity. This would be faster, cheaper, and more immediately beneficial for conservation than full de-extinction projects. Evolutionary rescue could leverage de-extinction technology in ways that provide immediate benefits for living endangered species. This represents a pragmatic approach to de-extinction technology, viewing it as a tool for conservation rather than an end in itself. Woolly mammoth genes for cold tolerance might be useful in Asian elephants facing climate change. Giant ground sloth genes for efficient digestion in harsh environments might help modern endangered herbivores. The genetic information, locked in extinct lineages, represents a vast library of evolutionary solutions to survival challenges. Using that information to help living species thrive might be the most practical application of de-extinction research. When a de-extinct animal is created and lives and breathes, fundamental questions immediately arise about what its legal status actually is. If a woolly mammoth is recreated, is it a wild animal subject to endangered species protections? Is it property that someone can own and control? Does it have legal rights as a living creature? The law has not caught up with de-extinction science, and there is no clear legal framework for what happens when a resurrected species enters the living world. Some legal scholars argue that a recreated animal should have the same protections as the original extinct species would have had under environmental law. Others argue that because the recreated animal is technically a genetic construct created by humans, it might be classified as intellectual property or as a genetically modified organism rather than a wild animal. This distinction matters enormously because it determines who can control the animal, what rights the animal has, and whether it can be exhibited, sold, or released into the wild. A woolly mammoth created by Colossal Biosciences exists in legal limbo. It is not technically the extinct species because the actual extinction happened thousands of years ago. The recreated animal is a genetic proxy, a living representation of extinct genes in the body of a modern organism. If a de-extinct animal is released into the wild, does it count as an invasive species or as a native species being restored? The answer affects whether it receives legal protection or whether it can be killed if it threatens other wildlife. The legal questions surrounding de-extinction are as complex as the scientific ones, and they remain largely unanswered. If Colossal Biosciences creates a woolly mammoth, who owns it? Does the company own it as a private asset? Does the government claim ownership? Or do the funding organizations that paid for the project own it? Colossal's current direwolves and other recreated animals are kept in private facilities, suggesting the company believes it owns the animals it creates. But if a de-extinct species is released into the wild, ownership becomes ambiguous and fraught with legal uncertainty. Most wild animals are not owned by anyone. They are public resources managed by government agencies on behalf of the public interest. But a recreated species that was created by a private company and funded by private investment might be claimed as intellectual property. The company could argue that it invested millions of dollars developing the technology and genetic sequences necessary to create the animal, and therefore has ownership rights to the result. This would be unprecedented in conservation law. No private company currently owns a wild species in the way that a company might claim ownership of a de-extinct organism. Indigenous peoples raise additional ownership questions that are equally complex. Many extinct animals were sacred to indigenous cultures and held profound spiritual significance. If a mammoth or passenger pigeon is recreated, do the indigenous peoples whose ancestors lived alongside these animals have ownership claims? Do they have the right to decide what happens to the recreated species? Some legal scholars argue that indigenous rights to cultural heritage might extend to recreated versions of animals that held cultural significance. But existing law does not clearly support this claim. The ownership question reveals how de-extinction fundamentally challenges existing property and conservation law frameworks in ways that are only beginning to be explored. The Xerces blue butterfly represents one of the most poignant extinction stories and one of the most relevant cases for understanding insect de-extinction. This butterfly was native to San Francisco and lived in coastal dunes near the city. In the early 20th century, the population was still relatively large, but as urban development expanded and dunes were converted to housing and gardens, the butterfly's habitat disappeared entirely. The last known Xerces blue was spotted in 1943. The species went extinct because its entire habitat was destroyed. Museum specimens preserved contain DNA that could be sequenced, and technology exists to identify the closest living butterfly species and edit it to recreate Xerces blue characteristics. But the butterfly's habitat no longer exists. The dunes near San Francisco have been entirely converted to developed land. There is nowhere to release a recreated Xerces blue that would provide the conditions it needs to survive and reproduce. The ecological challenge of recreating its habitat might be impossible or prohibitively expensive. The Xerces blue case demonstrates why habitat restoration might be just as important as genetic recreation for de-extinction to succeed. You cannot simply recreate an animal and expect it to survive if the world it evolved to live in no longer exists. Insect de-extinction presents unique challenges that differ fundamentally from mammalian de-extinction because insects undergo complete metamorphosis, transforming from larvae to adults through complex hormonal systems and developmental pathways. Editing an insect's genome to recreate extinct traits is theoretically possible, but understanding which genes control metamorphosis and how those genes interact is extraordinarily difficult. Insect development is compressed and rapid, happening in weeks or months rather than years. If genetic editing disrupts the developmental process, the insect might fail to develop properly or might produce sterile offspring. Scientists have successfully created genetically modified insects, like disease-resistant mosquitoes, designed to suppress disease transmission, but these modifications are typically single gene changes, affecting one trait. Recreating an extinct insect species would require editing multiple genes simultaneously while maintaining normal development and reproductive capacity. Insects also need specific temperature, humidity, and dietary conditions that must be precisely maintained in laboratory environments. They are disease vectors, which create significant biosafety concerns about creating new insect populations and preventing accidental release. Despite these challenges, insects play crucial roles in ecosystems, and extinct insect species recreated with restored ecological roles might help repair damaged ecosystems. But insect de-extinction remains primarily theoretical, with no active projects. Plant de-extinction has actually had more success than animal de-extinction because plants reproduce in different ways and require different genetic considerations. Some plants can be grown from preserved seeds. Some plants can be regrown from preserved tissue. Some plants can be recreated through backbreeding, selecting plants with traits similar to extinct species and gradually intensifying those traits through selective breeding over many generations. Scientists have successfully grown plants from seeds that have been preserved for decades or even centuries in museum collections and seed banks. The Arnold Arboretum in Boston maintains a seed bank with millions of plant seeds collected from around the world, with some seeds in the bank over 100 years old. When researchers germinate these ancient seeds and grow them into mature plants, they create living connections to plant populations that may no longer exist in the wild. Backbreeding has been used to recreate extinct varieties of crops and ornamental plants. By examining historical records and paintings, plant breeders have identified traits that were present in extinct plant varieties and bred modern plants to recreate those traits. The result is not a genetically identical plant, but rather a plant that resembles the extinct variety in visible characteristics. Plant de-extinction is generally less controversial than animal de-extinction because plants do not raise the same ethical concerns about consciousness and suffering. A recreated plant does not suffer or have feelings, and its existence does not pose ethical questions in the same way that a recreated mammal does. The Judean date palm represents one of the most successful and well-documented cases of plant de-extinction through seed germination. This palm tree was native to the Middle East and was highly valued by ancient peoples for its nutritious dates. The trees were mentioned in the Bible and in ancient Egyptian texts. Over time, the species was cut down for various purposes, and by the medieval period, it had become extinct in the wild. All that remained were references in historical texts and artwork. In the 1960s, archaeologists excavating ancient sites in Israel found preserved date palm seeds in sealed jars that had been stored for nearly 2,000 years. Scientists attempted to germinate these ancient seeds. Most were too damaged by time to grow, but eventually, one seed successfully sprouted. Scientists carefully nurtured the seedling, and it grew into a healthy palm tree. This tree, now known as Methuselah, became the oldest plant ever grown from an ancient seed. The success of regrowing the Judean date palm from 2,000-year-old seeds proved that plant genetic material could survive for extraordinarily long periods if preserved in the right conditions. More recently, additional ancient date palm seeds were successfully germinated, creating a small population. These trees represent a functional restoration of a species that had been extinct for hundreds of years. The case demonstrates that plant de-extinction can succeed without modern genetic engineering technology. Ancient seeds possess remarkable longevity under the right conditions because they have protective seed coats that prevent moisture loss and damage from external forces. The genetic material inside is in a dormant state, not actively metabolizing and degrading. This dormancy allows seeds to survive for years, decades, or even centuries without significant genetic degradation. Scientists have found viable seeds in Egyptian tombs, in museum collections, and in permafrost that are hundreds or even thousands of years old. The oldest known seed that was successfully germinated was a date palm seed that was approximately 2,000 years old. This extraordinary longevity occurs when seeds are stored in cool, dry conditions. Temperature is critical. Cold slows down all biological processes, including the degradation of genetic material. Dry conditions prevent microbial growth and chemical breakdown. Seeds stored in active seed banks are kept at temperatures near 0 Celsius and in conditions with very low humidity. Under these conditions, seeds can remain viable for decades and possibly centuries. If extinct plant seeds can be found, or if seeds from plants that are on the verge of extinction can be collected and preserved, future scientists might be able to grow these plants even after they go extinct in the wild. The challenge is that not all plants produce seeds that can survive long-term storage. Some seeds have short lifespans and lose viability within months. Scientists are working to understand the genetic basis of seed longevity so that they can develop preservation techniques for seeds from species with short natural lifespans. The ability to preserve plant genetic material in seeds makes plants fundamentally different from animals in their potential for de-extinction. Coral restoration and ancient reef reconstruction represents one of the most ambitious and urgent de-extinction-related projects currently underway. Coral reefs are not individual animals, but rather colonies of millions of tiny organisms living together in a complex ecosystem. When reefs are destroyed by warming water, pollution, or physical damage, the entire ecosystem collapses. Some coral species have gone functionally extinct in certain regions because the reefs they lived in no longer exist. Scientists are attempting to restore coral reefs by collecting surviving corals and breeding them in laboratory conditions to increase their numbers and genetic diversity. Some projects involve collecting coral larvae and nurturing them in controlled environments before transplanting them back to the reef. Others involve identifying heat-resistant coral varieties and breeding them selectively to create populations that might survive in warming oceans. The goal is to restore reef ecosystems that have been destroyed, which in a sense is de-extinction because it involves recreating populations of species that have disappeared from specific locations. Coral restoration differs from animal de-extinction in that it focuses on ecological restoration rather than genetic recreation. Scientists are not editing coral genes or attempting to recreate extinct coral species. Instead, they are trying to restore coral populations that currently exist but have been locally eliminated. Some coral reefs have been destroyed so completely that natural recovery is impossible without human intervention. Artificial reef structures have been built to provide substrate for coral larvae to settle and grow. These structures are seeded with coral larvae that are bred in laboratories. As the corals grow, they gradually build up the reef structure. Coral restoration projects represent a middle ground between pure conservation and true de-extinction, acknowledging that some ecosystems have been so damaged that passive protection is insufficient. Microbial de-extinction presents extraordinary challenges because microorganisms are invisible to the naked eye, and their genetic complexity differs fundamentally from that of larger organisms. A bacterium might have thousands of genes, each performing specific functions. Recreating an extinct microorganism would require understanding what every gene does and why it is necessary for the organism's survival and function. Scientists would need to synthesize an entire genome from scratch and introduce it into a living cell. This is theoretically possible, but extraordinarily difficult in practice. One theoretical application of microbial de-extinction is to recreate microorganisms that were responsible for producing specific compounds or performing specific ecological functions. Ancient soil microbes might have possessed genes for metabolizing compounds that modern microbes cannot process. However, there is significant concern about recreating extinct microorganisms because of biosafety risks. A recreated pathogenic microorganism could potentially cause disease. An extinct bacterium that was adapted to thrive in ancient environmental conditions might struggle in modern conditions and behave unpredictably. Scientists working on microbial de-extinction acknowledge that extreme caution would be necessary. Any recreated microorganism would need to be kept in secure laboratory facilities and extensively studied. No active microbial de-extinction projects are currently underway. Microbial de-extinction remains primarily a theoretical exercise. The passenger pigeon's social structure was fundamentally different from that of modern pigeons, and this difference matters enormously for any de-extinction project. Passenger pigeons were extremely social birds that lived in massive flocks. Historical accounts described flocks so large that they darkened the sky for hours. These flocks contained millions of birds flying together in coordinated patterns. The birds would descend on forests and consume seeds, nuts, and insects in overwhelming numbers. A single flock could devastate a forest's food resources, but the pigeons would then move on, allowing the forest to recover. This boom and bust feeding pattern was part of what maintained the forest ecosystem. Modern pigeons, like band-tailed pigeons, do not live in such massive flocks. They live in much smaller groups. The behavioral genetics of passenger pigeons involve traits related to flocking behavior, synchronization with other birds, and responding to signals from other flock members. These behavioral traits would need to be recreated through genetic editing if a de-extinct passenger pigeon population were to behave like historical passenger pigeons. But behavior is not fully determined by genetics. It is also learned from social experience. A passenger pigeon raised alone would not naturally form massive flocks. It would need to learn flocking behavior from other pigeons, and if it was raised with modern pigeons that do not flock in the historical way, it would learn modern pigeon behavior instead. Scientists planning the passenger pigeon de-extinction project understand this problem. They are discussing how they would teach recreated passenger pigeons to behave like their extinct ancestors. But the authenticity of such learned behaviors would always be questionable. Flock behavior requirements are one of the most challenging aspects of passenger pigeon restoration, because behavior was integral to the species' ecological role. Passenger pigeon flocks shaped entire forest ecosystems through their massive migrations and feeding patterns. When flocks moved through areas, they consumed enormous quantities of seeds, affecting which plants could reproduce. Some plants evolved seeds difficult to digest or too large to swallow. Migration routes followed seasonal food availability, creating natural disturbance that prevented certain species from becoming too dominant. The loss of passenger pigeon flocks changed forest composition. Without heavy seed predation, certain plant species became more abundant, cascading through herbivores and predators. If passenger pigeons are recreated, scientists hope to re-establish flocking behavior. But creating functional flocks of millions would require releasing hundreds of thousands of recreated birds simultaneously. There is no guarantee recreated pigeons would form massive historical flocks. They might form small flocks or scatter and live solitary lives. If flocking behavior cannot be recreated, ecological benefits would be severely limited. The ivory-billed woodpecker debate illustrates difficulty determining whether a species is truly extinct or simply extremely rare. This large woodpecker lived in southeastern US old-growth forests. The last confirmed sighting was in the 1940s. In 2004, researchers reported sounds and evidence suggesting ivory-billed woodpeckers might exist in remote Arkansas forests, sparking controversy. Some scientists said evidence was not definitive. Others argued a small undiscovered population might survive. Extensive searches found no definitive evidence. The species remains presumed extinct, though some maintain a tiny population might survive. The case raises important de-extinction questions. If a species is truly extinct, this must be definitively established before de-extinction efforts are justified. But how do you prove extinction when vast unexplored forests might hide populations? De-extinction projects focus on unquestionably extinct species with museum specimens and clear historical records. But the boundary between extinct and extremely rare, with small possibility of populations remaining, is fuzzy. The ivory-billed woodpecker remains in strange uncertainty that prevents de-extinction efforts. Extinction versus extreme rarity represents a philosophical and practical problem affecting how scientists prioritize conservation efforts. A species numbering fewer than 100 individuals is functionally extinct in that it will likely go extinct within decades due to inbreeding, but technically, it is still a living species. Should conservation focus on saving functionally extinct populations through genetic rescue and management, or focus on de-extinction of truly extinct species? The California condor provides a case study. This enormous vulture was hunted near extinction, reduced to 27 individuals by 1987. Through intensive breeding and reintroduction, over 500 exist today, though many in captivity. The Arabian Aurochs was hunted to extinction in the wild by the 197 zeros, with only a handful remaining in zoos. The Arabian Aurochs project bred them in captivity, and reintroduced them to protected Arabian areas. Today, hundreds exist in the wild. These examples show intensive management of functionally extinct populations can succeed. But each requires massive resources. If resources directed to de-extinction could recreate multiple extinct species, more total animals might be saved. The choice between saving dying populations and resurrecting extinct ones involves value judgments about which species matter most and how resources should be allocated. The Yangtze River dolphin search represents one of the most heartbreaking extinction stories and one with important lessons for de-extinction planning. The Baiji dolphin lived in the Yangtze River in China for millions of years. As recently as the 1980s, hundreds or even thousands were believed to exist. But the river ecosystem was devastated by shipping, dam construction, pollution, and fishing. The dolphin population declined rapidly through the 1980s and 1990s. By 2000, only a few dozen remained. In 2002, a year-long intensive search found no living Baiji dolphins. The species was declared functionally extinct, making it the first dolphin species driven to extinction in modern times by human activity. The tragedy of the Baiji was that the river habitat was killed before the dolphin population later recovered. Fishing nets continue to kill dolphins. Underwater noise from shipping disrupted their echolocation. Pollution accumulated in their tissues. The river ecosystem could not support dolphins anymore because it was so degraded. Even if Baiji dolphins could be recreated through de-extinction, the river they would need to live in no longer exists. Attempts have been made to search for dolphins that might still survive, with some reported sightings and alleged photographic evidence in recent years. But no definitive confirmation of a living Baiji has been found. Genetic material from preserved Baiji specimens exists and is being studied. Without habitat restoration, recreated dolphins would face the same extinction risk that eliminated the original population. The Baiji story definitely believed in the Russian population. The Baiji story definitely demonstrates why de-extinction without habitat restoration is incomplete. The fundamental problem was not the dolphin's genetics, but the environmental degradation that made survival impossible. Environmental DNA detection represents a new approach to finding supposedly extinct species. eDNA is genetic material organisms shed through scales, feathers, feces, urine, skin cells, and blood. When organisms live in water, their eDNA fills lakes, rivers, and oceans. Scientists collect water samples, filter eDNA, and use genetic sequencing to identify species present. This can detect species even when not directly observed. Scientists searching for Baiji dolphins used eDNA testing in the Yangtze River. No Baiji eDNA was detected, despite extensive sampling, suggesting true extinction. eDNA testing provides strong evidence of current presence, but weaker evidence of absence. A species could be extinct even without eDNA detection. However, repeated sampling over years with no detections suggests extinction is likely. eDNA technology is increasingly important in conservation biology because it monitors biodiversity without observing individual animals. For de-extinction projects, eDNA confirms species extinction before attempting recreation. Modern climate change impacts everything about de-extinction planning because the world that recreated animals would live in is fundamentally different from the world they evolved in. A woolly mammoth evolved in the Pleistocene Arctic, where temperatures were much colder and the landscape was a mixture of grasslands, tundra, and sparse forests. The modern Arctic is warming rapidly. The climate that suited mammoth physiology may not exist anymore in the places where mammoths might be released. Scientists working on the woolly mammoth de-extinction project have discussed introducing mammoths to rewilding areas in Siberia and potentially other Arctic regions. But the climate in Siberia is changing faster than almost anywhere else on Earth. Permafrost is melting, vegetation patterns are shifting, the Arctic ecosystem is being transformed. A recreated mammoth released into Siberia would face a climate regime that is different from anything its genetics are adapted to. The mammoth would still be genetically cold adapted, but the environment might be too warm. The ecological niche that mammoths occupied no longer exists as it once did. Climate change complicates de-extinction in another way. The resources required for de-extinction projects might be better spent on conservation efforts that help species adapt to climate change. De-extinction proponents argue that technology developed for de-extinction has applications for helping modern species adapt through genetic engineering and breeding programs. Will revived species survive in a radically transformed world remains the central question determining de-extinction success. A species is not just genetics, it is a web of relationships with environment, other species, food, nesting sites, predators, and competitors. When species go extinct, the entire web collapses. When recreated, the web must be reconstructed from scratch, possibly impossible at any meaningful scale. A single zoo mammoth represents no ecological restoration. A population released into Siberian rewilding areas represents something more significant. But mammoths would need to feed themselves, find appropriate food sources, and survive predation, disease, and competition. If the landscape lacks enough vegetation to feed hundreds of mammoths, the population would collapse. Scientists have calculated carrying capacities for mammoths in potential areas. Some estimates suggest Siberia might support thousands if suitable grasslands were maintained. But maintaining grasslands requires active human management. The landscape must be shaped to provide mammoth habitat. This differs fundamentally from true wild populations. True wild mammoths would be self-sustaining. Recreated populations might always require ongoing care. Whether this constitutes success depends on what success means. If success means creating animals that behave naturally and live independently, de-extinction might fail. If success means creating populations fulfilling ecological roles, even with human assistance, de-extinction might succeed. The moral obligation question remains at the heart of de-extinction debates because it forces society to confront difficult truths about human responsibility for extinction. Humans have caused most recent extinctions either directly through hunting or habitat destruction, or indirectly through ecosystem changes. If humanity caused the damage, does humanity have an obligation to fix it? Some philosophers and scientists argue yes, pointing to our newfound capability to undo extinctions through de-extinction technology. If we have the capability, and the species extinction was our fault, they argue we have a moral obligation to attempt restoration. But other philosophers argue that de-extinction does not actually restore what was lost. It creates genetic copies or approximations, but it does not resurrect the actual animals that lived and died. The passenger pigeons that were slaughtered in the 19th century are gone forever. Creating new pigeons with similar genetics does not bring back the individual birds that suffered. This perspective argues that there is something qualitatively different between restoration and resurrection. Some argue that the moral obligation we bear is not to bring back extinct animals, but to prevent current extinctions and to restore habitats where living species can thrive. Resources spent on de-extinction might be morally problematic if those resources could have prevented an extinction instead. This utilitarian calculation matters tremendously. De-extinction proponents counter that de-extinction funding often comes from billionaires and venture capital that might never have gone to traditional conservation efforts. In that sense, de-extinction funding is additive rather than competitive with conservation funding. Indigenous peoples and sacred species raise important questions about cultural preservation and representation in de-extinction projects. Many extinct species held profound cultural and spiritual significance to indigenous peoples. The passenger pigeon was important to Native American cultures. The Tasmanian tiger held significance to Aboriginal peoples of Australia. The dodo was known to indigenous Mauritians before European arrival. If recreated, what role do indigenous peoples play in deciding how animals are used and where they are released? Some de-extinction projects have begun working with indigenous communities to incorporate traditional knowledge. The South Island giant moa project involved Maori communities in New Zealand from the start, but in other projects, indigenous peoples undefed, were excluded, or had limited input. Who decides whether extinct species should be brought back? Is it purely scientific and economic, decided by scientists and billionaires, or do indigenous peoples, whose ancestors lived alongside these animals, have decision-making authority? These reflect broader power dynamics in conservation and research. Historically, conservation excluded indigenous peoples despite indigenous lands containing much remaining biodiversity. De-extinction projects offer opportunity to do better, including indigenous peoples as partners rather than research subjects. But legal frameworks for ensuring participation and benefit sharing remain underdeveloped. Genetic material comes from museums and archives, sometimes collected without indigenous consent. If recreated animals are created from this material, questions arise about indigenous compensation or ownership rights. Cultural significance of lost animals extends beyond indigenous peoples to include broader human societies with environmental and philosophical meanings. Many extinct animals have become symbols. The mammoth represents the Ice Age and lost prehistoric world. The passenger pigeon represents tragedy of overexploitation and human carelessness. The dodo represents vulnerability of island species to human interference. When we recreate these animals, we revive symbols, making real the creatures in our stories, art, and cultural memory. A recreated mammoth means something different than a museum skeleton. It would be a living presence representing previous epochs. For some, this would be inspiring. For others, it feels like appropriation of natural history or distraction from protecting living species. The cultural meaning of extinct animals affects whether de-extinction is pursued and how the public responds. If framed as restoration of what was lost, it might be viewed positively. If framed as hubris or playing God, it might be viewed negatively. These cultural and philosophical questions matter as much as scientific ones. De-extinction is about how humans understand nature, responsibility, and restoration. It is about what we value and what we believe we can and should do. Animals themselves exist within complex webs of human meaning and responsibility. Future technology possibilities suggest de-extinction might become easier, cheaper, and more refined over coming decades. Current projects like dire wolves and forthcoming mammoths are expensive and resource-intensive. Technology typically becomes more accessible and affordable as it matures. Gene editing is improving rapidly. Artificial reproduction systems are becoming more sophisticated. Scientists develop better ways to edit genomes with higher accuracy and fewer off-target effects. As technology improves, de-extinction might become feasible for more species with dramatically lower costs. Within decades, recreating dozens of extinct species at significantly lower costs might be possible. Synthetic biology is advancing where entire genomes can be designed and synthesized without actual format. Extinct DNA, complete genomes synthesized from databases, would eliminate need for museum specimens. Scientists could create organisms from pure genetic information, opening de-extinction to species with no surviving material, extinct millions of years ago. Alternatively, de-extinction might plateau and prove more difficult than current optimism suggests. Some extinct species might have genetic characteristics not replicable in modern organisms. Recreating behavioral repertoires might prove impossible. Ecological challenges of establishing populations in transformed landscapes might be insurmountable. De-extinction might remain limited to flagship species in managed environments. The future remains genuinely uncertain. What seems possible in theory might prove impossible in practice. Ultimately, de-extinction is not just a scientific question or a technical challenge that can be solved through better genetics and technology. It is a fundamental reflection of how humans understand ourselves and our relationship with the natural world. We have developed the capability to bring back extinct species through genetic engineering and reproductive technology. But whether we should undertake de-extinction, which species to prioritize, how to ensure these animals can survive and flourish in transformed ecosystems, and how to address the deeper conservation challenges that led to extinction in the first place, these are profound questions that require wisdom, humility, and careful moral thought. De-extinction offers hope that some of humanity's most devastating impacts on nature might be partially reversed. But it also risks creating a false sense of security, suggesting that conservation is unnecessary because we can always resurrect what we destroy. The most important lesson from de-extinction science is that living species deserve our protection and resources not because they might go extinct and need to be recreated later, but because they are valuable in themselves, right now, while they still live and breathe. The future of species preservation lies not just in laboratories and genetic engineering, but in protecting the habitats where animals still thrive, and in recognizing that the most effective conservation has always been prevention. De-extinction is a tool, remarkable and powerful, but it cannot replace the primary work of protecting life before it disappears forever into extinction. We must choose to value the living first.